100 Sleepy Facts About Rest and Recovery. Tonight we explore what happens inside the body when it finally stops moving. You've felt this before. The end of a long day, shoes off, the edge of the bed pressing against the back of your legs. You sit and then you lie back, and the mattress receives the full weight of your body for the first time in 16 hours. The blanket settles over your chest. Your shoulders drop a few millimeters into the pillow. The air near your face feels a little cooler than the air beneath the covers. And right there, in that quiet moment, something measurable begins to change. Your heart rate, which has been running between 70 and 90 beats per minute for most of the day, starts to slow. Not by much at first, maybe 5 beats less per minute within the first few minutes of lying down, but the direction is clear. Blood pressure drops. When you shift from standing to horizontal, gravity no longer pulls blood toward your feet. The baroreceptors in your carotid arteries register the change and send a signal to the brainstem. The brainstem responds by dialing back sympathetic nervous system activity. Your blood vessels relax. The work of circulating blood against gravity eases. Your breathing changes too. The respiratory rate, which averages 12 to 20 breaths per minute during the day, begins to settle toward the lower end of that range. Each breath becomes a little deeper, a little slower. These are not random events. They are the opening measures of a biological sequence that will unfold across the next several hours. Rest is not the absence of activity. It is a second kind of work, quieter, slower, carried out in the dark while you are barely aware it's happening. Over the next 2 hours, we'll move through 100 observations about what the body and brain do during rest, sleep, and recovery. Some of these observations come from decades of well-established research. Others come from newer studies still being tested and questioned. Where the evidence is strong, we'll say so. Where it's still developing, we'll say that too. The body you're resting in right now is running processes that took billions of years to develop. The impulse to close your eyes when the light fades is one of the oldest instructions written into living tissue on this planet. So let the weight of the day go. There's nothing you need to do right now. The first group of facts describes the architecture of sleep itself, the stages, the patterns, the electrical rhythms that cycle through the brain as the night moves forward. When you first begin to fall asleep, you enter a stage that researchers call N1. It is the lightest form of sleep. Your eyes may drift slowly beneath closed lids. Your muscles begin to relax, though not completely. If someone spoke your name, you would likely wake. The electroencephalogram, or EEG, a device that records electrical activity across the scalp, shows a shift during N1. The alpha waves that dominate quiet wakefulness, running at about 8 to 12 cycles per second, begin to break apart. They are replaced by lower amplitude mixed frequency activity. Theta waves, running at 4 to 7 cycles per second, appear in short bursts. N1 typically lasts between 1 and 7 minutes. Most people don't remember it. Some people experience a sensation of falling or a sudden muscle twitch called a hypnic jerk. This is common and harmless. The motor cortex fires a brief involuntary signal as the body transitions out of wakefulness. The second observation is about N2, the stage that follows. N2 is where you spend roughly half of a normal night's sleep. Heart rate slows further. Body temperature continues to drop. The brain becomes less responsive to external sounds and sensations. 2 distinct electrical patterns appear on the EEG during N2. The first is the sleep spindle, a brief burst of activity at about 12 to 16 cycles per second, lasting 1 to 2 seconds. Sleep spindles are generated by circuits connecting the thalamus and the cortex. The thalamus acts as a relay station for sensory information, and during N2, these spindle bursts help block incoming signals from reaching conscious awareness. You are being gently walled off from the outside world. The second EEG marker of N2 is the K complex. It appears as a brief, high-amplitude wave, a sharp downward deflection followed by a slower upward one. K-complexes can be triggered by external sounds, but they also occur spontaneously. Researchers believe they may help suppress cortical arousal, keeping you asleep even when there's noise nearby. From N2, the brain descends into N3. This is deep sleep, slow-wave sleep. It is the stage hardest to wake from and the one the body seems to need most urgently. During N3, the EEG shows large, rolling delta waves, slow oscillations running at about half a cycle to 2 cycles per second. These waves represent synchronized firing across large populations of cortical neurons. Millions of cells are switching between active and quiet states together, roughly once every second or two. This synchronized rhythm is not random. It serves a purpose. It creates conditions for metabolic clearing, for hormone release, for immune system signaling. The deeper facts ahead will describe these processes in detail. For now, the important point is that N3 sleep has a distinct electrical signature, and this signature appears most strongly in the first third of the night. After a period of deep N3, the brain reverses course. It moves back through lighter sleep and then enters a stage unlike any other. This is REM sleep, rapid eye movement sleep. During REM, the EEG looks different from the slow waves of N3. It shows fast, low-amplitude activity that resembles the pattern of an awake brain. Your eyes move quickly beneath closed lids. Your breathing becomes irregular. Heart rate fluctuates. But your body is still. During REM sleep, the brainstem sends inhibitory signals to the motor neurons of the spinal cord, producing a state called muscle atonia. Your large skeletal muscles are effectively paralyzed. You can dream of running, but your legs don't move. This paralysis is protective. Without it, you would physically act out the movements of your dreams. A small number of people have a condition called REM sleep behavior disorder, in which this inhibitory mechanism breaks down. They kick, punch, and shout during dreams, and this condition is now recognized as an early marker of certain neurodegenerative diseases. A single sleep cycle, the full sequence from N1 through N2, N3, and REM lasts roughly 90 minutes. In a typical 8-hour night, you move through 4 to 6 of these cycles. But the composition of each cycle shifts as the night progresses. The first 2 or 3 cycles contain the longest and deepest periods of N3. Your body front-loads deep sleep. By the last third of the night, N3 may nearly disappear from the cycle. In its place, REM periods grow longer. The final REM episode of the night, occurring in the early morning hours, can last 30 to 45 minutes. This pattern means the type of rest your brain gets changes across the night. Cut a night short by 2 hours, and you lose disproportionately more REM sleep. Go to bed 2 hours late, and you may lose a significant portion of your deep N3 sleep. The timing matters as much as the total duration. During NREM sleep, body temperature drops by 1 to 2 degrees Fahrenheit. The hypothalamus, a small structure deep in the brain, lowers its thermoregulatory set point. This is not a failure of temperature control, it is a deliberate adjustment. The body cools itself on purpose. This cooling appears to support the metabolic and restorative processes of deep sleep. It also means that sleeping in a cool room, around 65 degrees Fahrenheit, tends to align with what the body is already trying to do. A warm room works against the hypothalamus. These first 12 observations describe the basic architecture, the scaffolding that holds a night of sleep together. Stages, cycles, waves, and temperature shifts. Every night, whether you remember it or not, this sequence runs beneath your awareness like a program executing in the dark. A little farther inside the body, another pattern appears. This one involves fluid. The brain produces waste. Every cell does. Neurons, which consume more energy per unit mass than almost any other cell type, produce a steady stream of metabolic byproducts. During waking hours, these byproducts accumulate. In most organs, the lymphatic system handles waste removal. Lymphatic vessels carry cellular debris and excess fluid toward the bloodstream for filtering. But the brain doesn't have conventional lymphatic vessels inside its tissue. For a long time, scientists weren't sure how the brain cleared its waste at all. In 2012, a research team led by Macon Nettergaard at the University of Rochester published a study in mice that described what they called the glymphatic system. The name is a combination of glial, referring to glial cells, the support cells of the brain, and lymphatic. Here is what they observed. Cerebrospinal fluid, the clear liquid that surrounds and cushions the brain, flows into brain tissue along the outside surfaces of arteries. It moves through spaces around these blood vessels, called perivascular spaces. As it flows inward, it mixes with the interstitial fluid, the fluid that fills the gaps between brain cells. This mixing helps carry dissolved waste products outward toward veins, where the fluid drains away. In the mouse studies, this flow increased dramatically during sleep compared to wakefulness. The interstitial space between brain cells appeared to expand during sleep, making it easier for fluid to move. One widely cited estimate from this early work was a roughly 60% expansion of the extracellular space, though subsequent research has produced a range of figures, and the exact magnitude in humans is still being studied. A key player in this process is a protein called aquaporin-4, a water channel protein embedded in the membranes of astrocytes. Astrocytes are star-shaped glial cells that wrap their end feet around blood vessels throughout the brain. Aquaporin-4 channels on these end feet help direct the inflow of cerebrospinal fluid from the perivascular spaces into the surrounding tissue. In mouse models, when aquaporin-4 is disrupted or mislocalized, glymphatic clearance drops substantially. Whether this same mechanism operates in the human brain in exactly the same way is still an active area of research. Human neuroimaging studies have found evidence consistent with glymphatic transport, and some MRI techniques can track the movement of contrast agents through human brain tissue in ways that resemble glymphatic flow. But the direct cellular-level observations that have been made in mice are harder to replicate in living human subjects. The picture is becoming clearer, but it isn't complete. What researchers do know is that norepinephrine, a neurotransmitter associated with alertness and arousal, plays a role in regulating this system, at least in animal models. During wakefulness, norepinephrine levels are relatively high. During deep REM sleep, they drop. This drop relaxes the smooth muscle walls of small blood vessels in the brain, enabling slow, rhythmic pulses of blood vessel expansion and contraction. These pulses, called vasomotion, help drive cerebrospinal fluid through the tissue. When sleep is fragmented, interrupted by brief awakenings, noise, or breathing disruptions, norepinephrine doesn't stay low in the same sustained way. The rhythmic vascular pulses that support fluid transport become irregular. Research in animal models suggests that even when total sleep time stays the same, fragmented sleep reduces glymphatic clearance compared to consolidated, unbroken sleep. One of the waste products that the glymphatic system appears to clear is amyloid beta, a protein fragment that accumulates in the brains of people with Alzheimer's disease. In mouse studies, sleep deprivation increases amyloid beta levels in the brain, and chronic sleep disruption accelerates the formation of amyloid plaques. Human PET scan studies have also found that even a single night of sleep deprivation can increase amyloid beta accumulation in certain brain regions, though the long-term implications of occasional poor sleep versus chronic disruption are still being investigated. Tau protein, another molecule associated with neurodegeneration, also appears to be affected by sleep. Tau levels in cerebrospinal fluid rise during wakefulness and fall during sleep. In mouse models, sleep deprivation increases the spread of tau pathology through the brain. The evidence in humans is still developing, but the direction of the findings is consistent across species. Research suggests that the position of the body during sleep may influence glymphatic transport, though this finding is preliminary. A 2015 study in rodents, led by Hedok Lee and colleagues, found that side-lying positions appeared to enhance glymphatic clearance compared to sleeping on the back or stomach. Whether this translates directly to humans is not yet confirmed, but it has generated interest in the relationship between sleep posture and brain health. The glymphatic system also appears to be influenced by age. In older mice, glymphatic function declines. The aquaporin 4 channels on astrocyte in feet become less polarized, less concentrated at the perivascular surfaces where they're needed most. This may partly explain why sleep becomes lighter, more fragmented, and less restorative as people age. Alcohol, even in moderate amounts, has been shown to impair glymphatic function in animal studies. Low-dose chronic alcohol exposure reduces the polarization of aquaporin 4 and disrupts the coordinated vascular pulses that drive fluid transport. This is separate from alcohol's well-known effect of fragmenting sleep architecture. These 12 observations describe a cleaning system that appears to operate most efficiently in the deep, quiet hours of slow-wave sleep. The evidence is strongest in animal models and is being gradually extended to human brains through imaging studies and indirect measurements. The brain is not idle during rest. It is being washed, slowly and methodically, by a current of fluid that depends on the very stillness you feel right now. From here, the story moves a little deeper, into memory, and the way experiences are sorted and stored while you sleep. During the day, your brain encodes experiences into temporary storage. The hippocampus, a curved structure deep in the temporal lobe, acts as a kind of short-term holding area. Events, conversations, sequences of movement, they are written in the hippocampus first. But the hippocampus has limited capacity. It isn't meant to hold memories forever. During sleep, something happens. Memories begin to move. Sleep spindles, those 12 to 16 cycle bursts of activity that mark N2 sleep, play a role in this transfer. Studies using simultaneous EEG and functional MRI have shown that sleep spindle activity correlates with increased communication between the hippocampus and the neocortex, the large outer layer of the brain where long-term memories are stored. The spindle appears to open a window during which information can be relayed from one region to the other. At the same time, the hippocampus generates sharp-wave ripples. These are brief, high-frequency bursts of activity lasting 50 to 100 milliseconds. During a ripple, the hippocampus replays sequences of neural firing that occurred during the day. If you walked a specific route that afternoon, the neurons that fired in sequence as you walked may fire again in the same order during a sharp-wave ripple, but compressed into a tiny fraction of the original time. This replay is not random. It tends to prioritize experiences that were emotionally significant or that involved new learning. The brain is not replaying everything from the day. It is editing. The coordination between sleep spindles and sharp-wave ripples appears to be important. Studies have shown that when ripples in the hippocampus coincide with spindles in the cortex, memory consolidation is more effective. The two events lock together in a timing relationship, and disrupting that timing, even slightly, can impair the transfer of memories from temporary to long-term storage. REM sleep contributes to a different kind of memory processing. During REM, the amygdala, the brain's primary center for processing emotional significance, becomes highly active. Functional imaging studies show strong connectivity between the amygdala and the hippocampus during REM sleep. This interaction appears to help process emotional memories, though the exact mechanism is still being studied. One theory, supported by some evidence, suggests that REM sleep strips the emotional charge from difficult memories while preserving the factual content. You remember what happened, but it doesn't feel as raw. This process may be disrupted in post-traumatic stress disorder, where nightmares replay traumatic events without the emotional dampening that normally occurs during REM. Procedural memories, the kind involved in learning to play piano, ride a bicycle, or type without looking at the keyboard, appear to consolidate through a different pathway. These memories depend more on N2 sleep and the motor cortex than on hippocampal replay. Studies of motor skill learning have found that performance improves after a night of sleep, even without additional practice. The improvement correlates with the amount of N2 sleep obtained, and specifically with sleep spindle density over the motor cortex. This means that when you practice something new and then sleep, the sleep itself becomes part of the learning, not a metaphor, a measurable part. Working memory, the ability to hold and manipulate information in the short term, also appears to benefit from sleep. After sleep deprivation, working memory performance drops, and the decline correlates with reductions in prefrontal cortex activity. The prefrontal cortex is among the brain regions most sensitive to sleep loss. Researchers have tried to enhance memory consolidation during sleep using a technique called targeted memory reactivation. In these experiments, a specific sound or scent is paired with a learning task during the day. Then, during slow-wave sleep, the same sound or scent is presented at low volume. Studies have found that this can selectively strengthen the associated memories without waking the sleeper. The effect is modest but reproducible across multiple labs. Sleep also appears to help with creative problem-solving. A well-known study from 2004 by Ulrich Wagner and colleagues found that subjects who slept after learning a mathematical problem were more than twice as likely to discover a hidden shortcut compared to those who stayed awake for the same period. The insights seemed to emerge from the reorganization of information that happens during sleep. Not all memories benefit equally from sleep. Memories that a person expects to need later, those flagged by the brain as relevant, tend to be consolidated more strongly than memories considered unimportant. The brain doesn't just replay, it selects. These 12 facts describe sleep as a process of sorting, compressing, and rebuilding. The experiences of the day are not simply stored on a shelf. They are taken apart and reconstructed, filed into networks, linked to older knowledge, cleaned of irrelevant detail. While memory consolidation unfolds in the cortex and hippocampus, another set of processes runs in the endocrine system. The hormones of rest and repair begin to circulate. Growth hormone is released by the anterior pituitary gland in pulses throughout the day, but the largest pulse occurs during the first bout of deep N3 sleep. This typically happens within the first 90 minutes after you fall asleep. Research suggests that a substantial portion of the day's total growth hormone secretion, some estimates suggest more than half, occurs during this early window of slow-wave sleep. Growth hormone does more than support growth in children. In adults, it stimulates tissue repair, supports muscle protein synthesis, promotes fat metabolism, and aids bone remodeling. When deep sleep is selectively suppressed, by introducing noise that lightens sleep without fully waking the person, growth hormone secretion drops significantly. The hormone release is tied specifically to the slow-wave electrical pattern, not merely to being asleep. Cortisol follows a different rhythm. It is often called the body's primary stress hormone, but that label is incomplete. Cortisol is a glucocorticoid, a steroid hormone produced by the adrenal glands, and it plays a role in metabolism, immune function, blood sugar regulation, and dozens of other processes. Cortisol levels follow a 24-hour curve. They are lowest around midnight, when deep sleep is at its peak. They begin to rise in the early morning hours, climbing steeply in what's called the cortisol awakening response. By the time you open your eyes, cortisol levels have already increased substantially. This rise prepares the body for the metabolic demands of the waking day. If cortisol stays elevated at night due to stress, irregular schedules, or sleep disruption, it can interfere with the transition into deep sleep. High nocturnal cortisol is associated with lighter, more fragmented sleep and reduced slow-wave activity. The relationship runs in both directions. Poor sleep raises cortisol, and elevated cortisol impairs sleep. Melatonin is produced by the pineal gland, a small structure near the center of the brain. Its synthesis is controlled by light. During the day, light entering the eye travels along the retinohypothalamic tract, a direct neural pathway from the retina to the suprachiasmatic nucleus, the brain's central clock, and suppresses melatonin production. As evening light dims, that suppression lifts. Melatonin levels begin to rise roughly 2 hours before your habitual bedtime. Melatonin doesn't induce sleep the way a sedative does. Instead, it signals to the body that darkness has arrived. It lowers core body temperature slightly. It shifts the timing of various physiological processes. Think of it as a dusk signal, a chemical announcement that nighttime conditions are beginning. The timing of melatonin onset varies between individuals. Some people's melatonin begins to rise early in the evening. Others don't see a significant rise until later. This variation is part of what determines whether you're naturally inclined to go to bed early or stay up late, your chronotype. The underlying biology is genetic, driven by variations in clock genes like PER2 and PER3. Prolactin is another hormone that rises during sleep. It is released by the anterior pituitary gland and reaches its highest levels during the middle of the night. Prolactin supports immune cell maturation and function. It also plays a role in tissue repair and the regulation of inflammation. Sleep deprivation suppresses prolactin release, which may partly explain the immune vulnerability that follows poor sleep. Thyroid-stimulating hormone, or TSH, peaks in the late evening hours and decreases during sleep. The relationship between TSH and sleep is bidirectional. Thyroid disorders can disrupt sleep architecture, and chronic sleep disruption can alter thyroid hormone levels. The system is deeply interconnected. Insulin sensitivity follows a circadian pattern as well. The body processes glucose more efficiently in the morning than in the evening. During sleep, insulin secretion decreases as metabolic demand drops. But when sleep is disrupted or shortened, insulin sensitivity can decrease within just a few nights. Studies have shown that restricting sleep to 4 or 5 hours per night for less than a week produces measurable changes in glucose tolerance, changes that resemble early markers of metabolic dysfunction. Leptin and ghrelin, hormones that regulate appetite, are also affected by sleep. Leptin, which signals satiety, decreases after sleep restriction. Ghrelin, which signals hunger, increases. This combination helps explain why sleep-deprived people tend to eat more and crave higher-calorie foods. The body interprets insufficient sleep as a state requiring additional energy. Oxytocin, a hormone associated with social bonding and trust, has also been measured during sleep, though research in this area is less extensive. Some studies suggest that oxytocin levels may rise during certain stages of sleep and may contribute to feelings of calm, but the evidence is preliminary and not consistently replicated across studies. The hormone profile of a sleeping body is distinct from a waking one. Growth hormone up, cortisol down, melatonin circulating, prolactin rising. These shifts aren't side effects of sleep. They are core functions, chemical instructions that the body can only carry out when the brain enters certain states of rest. A little farther into the night, and a little farther into history, a different kind of observation appears. This one is about how human sleep has changed over centuries. For most of recorded history, nobody slept the way we do now. Or at least, not in the way that many people in industrialized societies sleep now. In the early 2000s, historian Roger Eckerch of Virginia Tech published research based on more than 500 references from pre-industrial European sources, diaries, court records, medical texts, literature, prayer manuals. Across these documents, Eckerch found repeated references to first sleep and second sleep. The pattern described was consistent. People went to bed not long after dark, slept for 3 to 4 hours, woke naturally around midnight, stayed awake for an hour or so, and then returned to sleep until dawn. The waking interval between first and second sleep was not treated as insomnia. It was expected. People use the time to pray, reflect, talk with a bed partner, or simply lie quietly in the dark. Some medical texts from the period recommended this interval as the best time for conception. It was ordinary life. Eckerch's research focused primarily on European sources between the 15th and 19th centuries. He found the pattern described in English, French, Italian, and other European languages. Whether this exact pattern was universal across all human cultures is harder to establish. The documentary evidence from other regions is less extensive, and sleep patterns likely varied with climate, latitude, and cultural practice. What is clear from Eckertsch's work is that the two-phase sleep pattern was widespread in the societies he studied, and it disappeared relatively quickly. The first major pressure came from artificial lighting. Gas lamps, introduced in European and American cities in the late 1700s and early 1800s, extended the usable evening. Coffee houses and social gatherings pushed later into the night. Bedtimes shifted. Then came electric light. Thomas Edison's incandescent bulb, commercialized in the 1880s, produced light far brighter and more sustained than any candle or oil lamp. Within a few decades, urban populations had access to reliable artificial illumination well into the night. The gap between first and second sleep compressed. By the early 20th century, references to segmented sleep had largely vanished from European and American texts. Eckertsch proposed a mechanism for this transition. As bedtimes moved later but wake times remained anchored to work schedules, total sleep opportunity shrank. The resulting increase in sleep pressure, the homeostatic drive to sleep, may have produced deeper, more continuous slow-wave-rich sleep that consolidated into a single block. The waking interval that once naturally separated two periods of sleep was squeezed out. The consolidation of sleep into a single block of roughly 8 hours is a historically recent development. In widespread practice, it is only about 150 years old. This doesn't mean it's unhealthy. Humans are adaptive, and the consolidated pattern may work well for most people. But it does mean that waking briefly in the middle of the night is not necessarily a disorder. It may be an echo of an older rhythm. Some researchers have tested this experimentally. In the early 1990s, psychiatrist Thomas Weyer at the National Institute of Mental Health placed volunteers in conditions that mimicked pre-industrial light exposure, 14 hours of darkness per night, with no artificial light. After several weeks, the subjects spontaneously began sleeping in two distinct bouts, separated by a quiet waking period of 1 to 2 hours. Their hormonal profiles during the waking intervals showed elevated prolactin levels, a pattern associated with calm, meditative states. Contemporary hunter-gatherer populations, studied with actigraphy, wrist-worn motion sensors that track sleep patterns, do not consistently show biphasic sleep. A 2015 study by Jerome Siegel and colleagues, examining sleep in 3 traditional societies in Tanzania, Namibia, and Bolivia, found that sleep was generally consolidated into a single period. Average sleep duration was 6 to 7 hours. These findings complicate the idea that biphasic sleep is the natural human default and suggest that sleep patterns vary with environmental and cultural context. What seems most reliable from the evidence is that human sleep is flexible. It can adapt to different light environments, social structures, and metabolic demands. The 8-hour consolidated block is one pattern among several that humans have used. It is recent, but it is not necessarily wrong. The key insight from this historical perspective is that sleep is shaped by environment as much as biology. The architecture of sleep, its stages, its cycles, its timing responds to the world around it. From the history of sleep, we return to the living body. The next set of facts looks at what happens in cells and in the immune system while you rest. Your immune system does not shut down during sleep. It reorganizes. T cells are a critical component of adaptive immunity. They identify and destroy infected cells. During sleep, the ability of T cells to adhere to their targets increases. This happens through a mechanism involving integrins, sticky proteins on the surface of T cells that help them lock onto infected cells. During waking hours, stress-related signaling molecules like adrenaline and prostaglandins partially suppress integrin activation. When you sleep, the levels of these molecules drop. With less suppression, the integrins become more active. T cells grip their targets more effectively. A study published in the Journal of Experimental Medicine in 2019 by Stoyan Dimitrov and colleagues demonstrated this mechanism using blood samples from sleeping and waking volunteers. The difference was measurable within a few hours of sleep onset. Cytokines, small signaling proteins that coordinate immune responses, also shift during sleep. Interleukin-1 and tumor necrosis factor, both pro-inflammatory cytokines, increase during nighttime sleep. This may sound counterproductive, but controlled inflammation is a necessary part of tissue repair and pathogen defense. The immune system uses the relative safety of sleep to run maintenance processes that would be disruptive during active waking hours. Sleep deprivation blunts this immune upregulation. Studies have shown that people who sleep fewer than 6 hours per night in the week before receiving a flu vaccine produce fewer antibodies compared to those who sleep 7 hours or more. The immune system needs sleep to mount an effective response to new threats. One study that generated significant attention was a 2015 paper by Eric Prather and colleagues, published in the journal Sleep, which tracked the sleep habits of 164 volunteers who were then exposed to a common cold virus. Those sleeping fewer than 6 hours per night were substantially more likely to develop a cold than those sleeping 7 hours or more. The specific risk ratio varied by analysis method, but the direction of the effect was clear and statistically significant. In a different line of research, scientists at Bar Ilan University in Israel studied chromosome dynamics in zebrafish neurons. Using fluorescent markers attached to chromosomal loci, they tracked the physical movement of DNA within individual neurons in real time. They found that during sleep, chromosomes moved more, increasing the activity of DNA repair enzymes at sites of accumulated damage. During wakefulness, these repair processes slowed. The researchers proposed that sleep provides a window during which neurons can address the DNA damage that builds up during the metabolic demands of waking hours. Whether this exact mechanism operates in mammalian brains in the same way is not yet established, but the principle that cellular repair processes are more active during sleep is consistent with findings across multiple species and cell types. Wound healing is also affected by sleep. Studies in both animals and humans have found that sleep deprivation slows the rate of skin wound recovery. One study published in the Journal of Applied Physiology found that sleep-restricted subjects showed meaningfully delayed wound healing compared to well-rested controls. The exact rate of delay varied between studies, but the pattern was consistent. Muscle protein synthesis, the process by which damaged muscle fibers are rebuilt after exercise, increases during sleep when amino acids are available in the bloodstream. This is why post-exercise nutrition and sleep are both considered important for recovery. The repair process depends on the raw materials being present and on the hormonal conditions, particularly elevated growth hormone, that sleep provides. Bone remodeling follows a circadian pattern as well. Osteoblasts, the cells that build new bone, and osteoclasts, the cells that break down old bone, operate on different schedules. Bone formation markers tend to peak at night. Chronic sleep disruption has been associated with decreased bone mineral density in some epidemiological studies, though establishing a direct causal link in humans requires more research. The gut microbiome, the trillions of bacteria living in the digestive tract, also appears to be influenced by sleep patterns. Studies in both mice and humans have found that sleep disruption alters the composition of gut bacteria, shifting the balance toward species associated with inflammation and metabolic dysfunction. The gut-brain axis, a bidirectional communication system between the digestive tract and the central nervous system, means that these changes can influence mood, appetite, and immune function. Natural killer cells, another component of the innate immune system, show reduced activity after sleep deprivation. These cells patrol the bloodstream and destroy cells that have been infected by viruses or that show early signs of becoming cancerous. Their activity drops measurably after even a single night of restricted sleep and recovers after a full night of rest. The immune system treats sleep as a maintenance window. It upregulates repair processes, strengthens defensive responses, and clears accumulated damage. When that window is consistently shortened or fragmented, the system falls behind. The body in front of this narration, your body, is running these processes right now if you'll let it. The T cells are gripping tighter. The cytokines are circulating. The chromosomes are being checked for damage. A little closer to the present day, and a little closer to the habits of modern life. The next set of facts looks at how the environment of the 21st century interacts with these ancient systems. The screens we look at in the evening emit light. All light suppresses melatonin to some degree, but not all wavelengths are equally effective. The retina contains a specialized class of cells called intrinsically photosensitive retinal ganglion cells, IPRGC for short. These are not the rods and cones that create visual images. They are cells that measure the brightness and spectral composition of light and send that information directly to the suprachiasmatic nucleus, the brain's central clock. IPRGC are most sensitive to light in the blue portion of the spectrum, wavelengths between roughly 460 and 480 nanometers. The screens of phones, tablets, and laptops emit a significant proportion of light in this range. When this light reaches the IPRGC in the evening, it sends a signal to the suprachiasmatic nucleus that resembles daylight. Melatonin production is suppressed or delayed. A 2014 study by Anne-Marie Chang and colleagues, published in the Proceedings of the National Academy of Sciences, compared the effects of reading on a light-emitting e-reader versus a printed book in the hours before bed. The e-reader group showed suppressed melatonin levels, delayed melatonin onset, reduced evening sleepiness, and delayed circadian timing. They also had less REM sleep and reported feeling more tired the next morning, even after 8 hours in bed. The effect is not just about the blue wavelength. Intensity matters too. A dim screen held at arm's length produces less melatonin suppression than a bright one. The timing of exposure is also important. The same screen that causes significant melatonin suppression at 10 p.m. may have little effect at 3 p.m. because the circadian system is less sensitive to light during the biological day. Core body temperature rhythms can shift with evening light exposure. Studies have measured delays of 30 to 60 minutes in the temperature minimum, the lowest point in the body's daily temperature cycle, after evenings spent in front of bright screens. Since the temperature minimum is linked to circadian phase, this shift can push the entire sleep-wake cycle later, without the person noticing any change in how sleepy they feel at bedtime. Chronic partial sleep restriction, consistently sleeping 6 hours instead of 7 or 8, produces effects that accumulate over time. A landmark study by Hans van Dongen and colleagues, published in 2003 in the journal Sleep, followed subjects restricted to 4, 6, or 8 hours of time in bed for 14 consecutive nights. The 6-hour group showed progressive declines in cognitive performance across the 2 weeks. By day 14, their reaction times and attention lapses were comparable to those of subjects who had been kept awake for an entire 24-hour period. Here is the part that caught the attention of researchers. The 6-hour sleepers did not rate themselves as particularly impaired. They reported feeling only slightly more tired than usual. The gap between self-assessed alertness and measured performance widened steadily over the 2 weeks. In other words, chronic sleep restriction impairs the ability to recognize that you are impaired. You adapt to the feeling of tiredness while the actual cognitive deficit continues to grow. Social jet lag is a term coined by sleep researcher Till Ronneberg. It refers to the difference between your sleep timing on work days and your sleep timing on free days. If you go to bed at midnight and wake at 7 on work days, but go to bed at 2 and wake at 10 on weekends, your social jet lag is roughly 2 hours. Your biological clock is being shifted back and forth each week. Epidemiological studies have found that greater social jet lag correlates with higher body mass index and with increased levels of inflammatory markers, independent of how much total sleep a person gets. The misalignment between the internal clock and the external schedule appears to carry metabolic costs of its own. Noise is another disruptor. Even sounds that don't fully wake you can trigger brief cortical arousals, moments where the brain shifts briefly toward a lighter sleep stage before returning to deeper sleep. These microarousals fragment the continuity of sleep without leaving any memory of waking. People who live near highways or under flight paths often show increased rates of cardiovascular disease, and chronic nighttime noise exposure is one proposed mechanism. Caffeine has a half-life of roughly 5 to 6 hours in most adults. A cup of coffee at 4 p.m. still has about half its caffeine in your system at 10 p.m. Caffeine blocks adenosine receptors in the brain. Adenosine is a molecule that builds up during waking hours and promotes sleep pressure, the feeling of drowsiness that increases the longer you stay awake. By blocking adenosine receptors, caffeine masks the feeling of sleepiness without reducing the underlying need for sleep. Alcohol is sometimes used as a sleep aid, but its effects on sleep architecture are disruptive. Alcohol increases deep sleep in the first half of the night, but fragments sleep in the second half, reducing REM sleep and increasing awakenings. It also relaxes the muscles of the upper airway, worsening sleep-disordered breathing in people prone to snoring or apnea. The net effect of alcohol on recovery sleep is negative, even when it helps with initial sleep onset. Temperature in the sleep environment affects sleep quality in measurable ways. The body needs to cool during the night, and a room that is too warm interferes with this process. Studies have found that a bedroom temperature between roughly 60 and 67 degrees Fahrenheit supports the best sleep for most adults. Humidity also matters. Excessively dry or humid air can increase awakenings. Exercise timing affects sleep, but the relationship is more nuanced than commonly believed. Morning and afternoon exercise generally improve sleep quality and reduce the time it takes to fall asleep. Evening exercise, depending on its intensity and how close to bedtime it occurs, may or may not disrupt sleep. A 2018 meta-analysis published in Sports Medicine found that vigorous exercise ending more than 1 hour before bedtime did not generally impair sleep, and in some cases improved it. These facts describe a modern environment that is fundamentally different from the one human sleep systems evolved in. Bright screens, caffeine, irregular schedules, background noise. Each one interacts with biological systems that were calibrated for a world of firelit evenings and consistent sunrise-wake times. The mismatch is not catastrophic, but it is real, and it adds up. The body can adapt to some of these pressures. It cannot adapt to all of them at once, not without cost. From here, we move toward recovery itself. What happens when the body gets the chance to make up for lost sleep, and whether that's even possible? Sleep banking is a concept that has gained attention in recent years, particularly in military and shift work research. The idea is straightforward. If you know that a period of sleep deprivation is coming, a long shift, a deployment, a string of early flights, can you store extra sleep in advance and use it as a buffer? A 2024 review of military sleep research examined the evidence for preemptive sleep extension, sleeping more than usual in the days before a period of expected deprivation. The findings were mixed but leaned in a positive direction. Extended sleep before deprivation did appear to protect sustained attention and reaction time during the first 24 to 30 hours of subsequent sleep loss. Subjects who had banked extra sleep performed better on vigilance tasks, particularly the psychomotor vigilance task, a standard measure of reaction time and attention lapses, compared to those who entered deprivation with normal sleep. But the protection was selective. Sleep banking showed weaker and less consistent effects on higher-order cognitive functions. Executive decision-making, working memory, and complex problem-solving did not benefit as reliably from preemptive sleep extension. The extra sleep seemed to protect the simpler, more automatic aspects of alertness while leaving the more demanding cognitive processes vulnerable. How much extra sleep makes a difference? The studies varied, but even modest extensions, 1 to 2 extra hours per night for several nights, produced measurable improvements in morning reaction time and reduced attention lapses. You don't need to sleep 12 hours a night to build a buffer. Small, consistent additions to sleep duration appear to accumulate. Recovery sleep after deprivation follows a predictable pattern. The brain prioritizes deep, slow-wave sleep first. During the first recovery night after sleep loss, the proportion of N3 sleep increases substantially, often at the expense of lighter sleep stages. This is called slow-wave rebound. The brain acts as though it has a deficit to repay, and it starts with the deepest, most restorative stage. REM rebound occurs later. If deprivation continues for more than 1 or 2 nights, the recovery process may take several nights to fully restore normal sleep architecture. REM sleep proportions gradually return to baseline, but the process is slower than the N3 rebound. One important finding from recovery sleep research is that the clock does not reset cleanly. After several nights of restricted sleep, a single night of extended sleep improves performance substantially, but it may not return performance to the pre-deprivation baseline. The debt appears to be partially but not fully repaid. A study by Daniel Cohen and colleagues at Brigham and Women's Hospital found that chronic sleep restriction creates a cumulative cognitive deficit that cannot be fully reversed by a single extended sleep opportunity. Subjects who had been restricted to 5.5 hours per night for 3 weeks required more than one recovery night to return to baseline performance on attention tasks. The longer the restriction period, the longer the recovery period needed. Napping, as a form of partial recovery, has been studied extensively. A nap of 20 to 30 minutes, short enough to avoid entering deep N3 sleep, can improve alertness, mood, and reaction time for 1 to 3 hours afterward. This benefit is well established across multiple studies and populations. Longer naps, in the range of 60 to 90 minutes, allow entry into deeper sleep stages and can produce more substantial cognitive benefits. But they also carry a higher risk of sleep inertia, the grogginess and confusion that sometimes follows waking from deep sleep. Sleep inertia typically dissipates within 15 to 30 minutes but can last longer, depending on the depth of sleep reached. The timing of a nap matters. Early afternoon naps, taken during the natural circadian dip in alertness that occurs roughly 6 to 8 hours after waking, tend to be most effective and least disruptive to nighttime sleep. Late afternoon or evening naps can delay sleep onset at night and create a cycle of fragmented nighttime rest. For shift workers, strategic napping before a night shift has been shown to reduce errors and improve vigilance during the shift. Some emergency medicine programs now include protected nap opportunities for physicians working overnight. The practice is still not widespread, but the evidence supporting it continues to grow. Recovery is not just about sleep duration. Sleep quality, continuity, depth, and appropriate cycling through stages matters at least as much. 8 hours of fragmented sleep with frequent awakenings may leave someone feeling less restored than 6 hours of consolidated, unbroken sleep. The architecture of the night matters as much as its length. And now, for these last observations, the perspective widens. Sleep is not a human invention. Nearly every animal with a nervous system shows some form of rest activity cycling. Fruit flies sleep. They become inactive, they adopt a specific posture, and they are harder to arouse. If you keep a fruit fly awake, it sleeps longer afterward, a pattern of homeostatic rebound that mirrors what happens in humans. The genes that regulate sleep in fruit flies have close equivalents in the human genome. Octopuses cycle between two distinct states that resemble quiet sleep and active sleep. During active sleep, their skin flickers with color changes, the same chromatophore patterns they use during waking behavior. Some researchers have speculated that octopuses may experience something like dreaming, though this remains unproven. The brain of an octopus is organized very differently from a vertebrate brain, yet the alternation between sleep states is present. Jellyfish, which have no brain at all, only a distributed nerve net, show sleep-like behavior. In a 2017 study published in Current Biology, researchers demonstrated that the upside-down jellyfish Cassiopeia reduces its pulsing activity at night, responds more slowly to stimuli, and shows a rebound in quiescence after being kept awake. This suggests that some form of sleep may not require a brain. A nervous system, even a simple one, appears to be enough. Nematodes, tiny roundworms with only 302 neurons, also exhibit a sleep-like state called lethargus. During lethargus, the worm becomes inactive and shows reduced responsiveness to touch. The state is regulated by some of the same molecular pathways that control sleep in mammals. The circadian clock, the internal timer that synchronizes biological processes to the 24-hour rotation of the Earth, is older than any of these animals. Cyanobacteria, Single-celled organisms that have existed for at least 2.5 billion years possess a molecular clock made of just 3 proteins: Chi-A, Chi-B, and Chi-C. These proteins cycle through phosphorylation states on a roughly 24-hour rhythm. They do this even in a test tube without any external signal. The timer is built into the chemistry itself. This means that the circadian rhythm predates multicellular life. It predates eyes. It predates brains and bones and blood. The rotation of the Earth has been imposing a cycle of light and darkness on living systems for so long that the response to that cycle is written into some of the most ancient molecular machinery on the planet. The planet that turns beneath you tonight has been doing so for 4.5 billion years. Long before there were forests, before there were oceans in their current form, before any creature opened an eye, the Earth was already cycling through light and dark. And living things, even the simplest, smallest living things, were already responding. When you feel your eyelids grow heavy as the room darkens, you are participating in a rhythm that is not just old. It is one of the oldest continuous biological processes still running on Earth. The body you are resting in carries instructions refined across deep time. Instructions to slow down when the light fades, to let the muscles go quiet, to let the temperature drop, to let the fluid circulate through the spaces between neurons, to let the hormones shift, to let the immune system do its quiet, careful work. Rest is not something you have to earn. It is something the body already knows how to do. It has known for a very long time. The cells are running their repair sequences. The hippocampus is sorting through the day. The slow waves are building, one cycle at a time. You don't need to follow any of this. You don't need to remember it. The body will carry on without your attention, just as it has every night you've ever slept. The Earth turns, the darkness holds, and somewhere inside you an ancient rhythm continues its work, patient, steady, older than nearly everything you've ever known. Let it carry you.